Well, thank you very much for that very nice introduction. I have to say that I'm completely flattered to have won this prize. There are many people sitting in the audience who do just as much of this kind of work as I do, and I think I'm sort of very lucky to be the one who's got the prize this year. I think part of that luck comes down to the virus, which I chose to work on because it lends itself to communication on a regular basis, being a virus that emerges both as a pandemic virus and then stays with us year on year as seasonal flu, and is always in the news and is familiar to the general public, so that everybody has an opinion about this virus which they like to offer up. And sometimes it's important to perhaps correct some of those opinions or home remedies or theories and make sure that we really are getting the science across. So it is a wonderful opportunity working on a virus like flu to sort of be able to both comment on how viruses evolve and how that impacts on everyday life, but also perhaps introduce a little bit of basic virology into to the readers of the Daily Mail and the listeners of Sky News. I think, from my point of view, I really sort of started doing this more frequently during the swine flu pandemic in 2009, which was the first, of course, pandemic of the 21st century. And I hope to tell you a little bit more about my experiences in doing that as we go along. But just sort of to bring us right up to date, you may know, as I've made the point already, this pandemic virus is still with us today, called now a seasonal flu, H1N1 virus, and many of you in the audience may actually have experienced it recently. There seems to be rather a large outbreak of it, unusually late this year, and so I suspect that we have more talking to do in explaining to the public exactly what's going on at the moment with this virus. I also am extremely lucky to have been supported by some wonderful people who have either worked in my group or been my collaborators or mentors, and I want to just acknowledge them on this slide, both present group members, past group members, some of whom have stayed in the flu area, which is very useful because they are sort of my insiders, people who work now at Public Health England, at NIBSC, who really have their fingers on the pulses of what's going on with this virus on an everyday basis, and who are, perhaps because they feel somewhat obliged to, are able to communicate to me as a mere academic what's actually going on out there in the real world. And also a whole list of colleagues who are fantastic to talk to about the virus as it evolves and about virology in general, and sort of to shape one's opinions and sort of view of that. So I just wanted to say thank you very much to all of those people and many more of you in the audience who have sort of moulded me into the communicating virologist that I am. This is all right. Let's go for that. Okay, and then the other reason why I am lucky to be here, as opposed to many other people in the audience who do this kind of thing, is that I work at Imperial College in London. Imperial is fantastically supportive of science communication. I think it's a really fantastic example of a university institute that has gone out there and encouraged its scientists to appear on the television, in the media, to make comment on science in the real world. And for that, I'm very grateful. And of course, it sits in the middle of London, which is not too far from the BBC studios and the Millbank studios. And so I think it is very useful to be able to work in London when one's working in this kind of area, and to shoot across in a taxi from one studio to the next. So I remember a particular day in the swine flu pandemic in around about May or June 2009, when there was a lot to be said, doing a string of interviews at different studios across London, jumping in taxis from one to the next. It was wonderfully exciting. I must say it's really invigorating to do that, but again, to give due thanks to the people in my group who were back in the office searching ProMed mail, emailing me regular updates to be picked up on my iPhone just before I went on live TV to sort of make a comment that was at least backed up by some sense of the most current facts. And Imperial College, being such a great place for science communication, encourages science communication right from the very early days. So to give one example, third-year undergraduates who are reading biochemistry, for example, have the option of choosing to do a science communication course in their final year alongside their literature-based projects. And this actually was pioneered by a previous Peter Wildy Prize lecturer, Stephen Curry. So he now runs this science communication course and pulls in all kinds of people with experience in the media, journalists, to encourage those undergraduate students to think about how to communicate science, how to get messages across, and the importance of various controversial issues. And in fact, I teach a particular class on that course where we discuss the gain-of-function controversy, which we're going to talk about in a minute, and the students at the end of it get to interview me in a mock-up radio interview in which they've only got 3 minutes to make me say the right things, and it's actually really quite a good, I think, experience for them to realise that getting a message across in a very compact space of time is quite an art, but also that from the other side of the questioning table, asking the right questions is very important. And I think that's true both for science and for communication. The other thing at Imperial is that there are master's courses in science communication. And so people who are perhaps scientifically trained, but are thinking that they're not going to stay in the lab, but want to use their science and do something more media-orientated, can come and study towards one of these master's degrees. And those master students are encouraged very much to input into the Imperial College magazine, iScience. And as a result of that, I was lucky enough to meet earlier this year a young man who's doing that master's degree at the moment, Jamie Brown. And for his sins, because I agreed to be on his programme, if you like, I have persuaded Jamie to come here today to interview me. So rather than me standing here anymore and talking to you in this sort of lecture format, what we're going to do now is we're going to move across to the hot seats, and Jamie is going to take over from here and just sort of give you, I suppose, a flavour of the sorts of things that I get to talk about pertaining to flu, and hopefully draw out from me some current thoughts and some current issues, as well as some past issues that I've had the honour to contribute to in various forms of the media.
As you've become this highly proactive science communicator, I also know that coincidentally you're quite interested in flu. So how do the two marry together in terms of having a disease, a virus, which everyone's very familiar with in the public? How do you have to handle that? Does it throw up challenges that everyone has an opinion?
Yeah, okay, so I mean, one of the real problems with working on flu is that there are a lot of fallacies out there that one needs to correct in a sort of informed way. Invariably, if at the end of spending a lunchtime giving a talk, somebody will come up to me and say, well, you see, I never get the flu because I boil an onion and inhale the juices every night. And then one needs to smile politely and sort of explain that perhaps there are other reasons why they're not being infected. So it is a virus which is very familiar to everybody, and that has its bad points, but it also has its good points because people are genuinely interested because it impacts directly on them. They all know somebody who's had flu, or they've had it themselves, or they have a story to tell. So you can grab people's attention by making it so real to them.
Yeah, absolutely. And so I suppose you've highlighted some of the occasions you've been called into action. Which have been the key stories and the key events? Maybe if you go back to your first sort of interaction with the media that you were called out and involved in.
Sure. So I think all the way through the early part of the 21st century, people have been very conscious of a rising awareness of the threat of pandemic flu, particularly from Asia, where various strains of flu appeared to be crossing unusually into humans and causing some alarm. The most famous one of those is the H5N1 bird flu, and that virus has been featured in magazines and newspaper articles, etc. And one of the sort of major things that I've been called on to do is to try to help use my knowledge to tell the public about the risks of such viruses and how we go about risk assessing those. So all flu viruses are coming from birds. Wild birds are the natural reservoir of flu. Occasionally, those viruses undergo various mutations and cross into people and can cause outbreaks that we call pandemics. And what people really want to know is how likely is that one of these pandemics is going to come along next time. As a result of a whole bunch of research that's gone on through the last decades, we understand that there are several barriers, thankfully, that protect us from regularly getting infected by avian viruses. For example, the virus doesn't bind very well to the human types of receptors if it's been evolving in birds. And so that's one change that has to happen. Also, the polymerase of the virus, which amplifies it in the host, doesn't work very well if it's been evolving in birds, and it has to undergo some other mutations to help transform it into a virus that can replicate productively in humans. And so all of these things sort of save us, if you like, from the pandemics that may have arisen from the H5N1 type of viruses or the H7N9, which is a more recently reported and worrying avian virus. Occasionally, these kinds of bird flu viruses, of course, come along in the UK, and then it's all much closer to home. Then the British media are much more excited about that, and I find myself being called upon to sort of comment on whether or not the latest outbreak in a particular poultry farm or an area of the country is truly going to pose a threat. Some people are more concerned about the chickens and turkeys. Are there going to be enough turkeys left for Christmas, for example, is a question I have been asked. But usually what's important to do is to explain to people that there are these barriers which, thankfully protect us as humans from ready infection by avian influenza viruses, and that it's important to understand the nature of those barriers and how far away any one particular virus is from being able to jump across and overcome those hurdles. Sure.
And I mean, around that time, was that leading towards the issues around the gain of function studies that were happening at that sort of time?
Absolutely. So back in sort of the early 2000s, a lot of people were trying to understand this very question, whether the H5N1 bird flu and now whether the H7N9 bird flu are going to transform into viruses which can not only infect humans by using the right receptors and replicate in our cells, but also transmit through the air from person to person. And in efforts to answer that question, scientists were using a combination, actually, of genetic engineering of viruses and also what we might call forward genetics, passaging viruses under certain selective conditions, to ask whether or not these H5N1 strains could indeed adapt to become transmissible between humans. And in doing that research, they sparked what is now known, I suppose, as the gain of function controversy, which I have been quite involved in explaining the background to, but also in sort of talking to the media about.
Okay. And I mean, in terms of like communication, is there any issues where there's the kind of thing that people could maybe be involved in earlier on? Is it maybe just because it sounds like a scary idea that people are playing with these viruses and like examining how far they could advance. So how would you possibly go about allaying those kind of fears?
Well, that's right. I think you're right in that the lay public do find that scary. And I think the problem with the way the gain of research work was rolled out was that it was perhaps not communicated very sensibly in a strategic sort of way. So people heard about this sort of outbreak, or not this outbreak, this discovery that the virus could pass between ferrets, which are the model for human-to-human transmission, at a conference where somebody stood up and said, hey, we've made this virus, isn't it great? And various members of the audience were somewhat alarmed, as you might be if you were hearing about this for the first time. Of course, what one has to do with hindsight is to explain that first of all, this is really quite a standard practice for virology and for this kind of research in general, this sort of combination of forward and reverse genetics, the dissection of different phenotypes by using this kind of mutational approach has been going on for many years. Secondly, of course, all of these experiments are performed in appropriate containment laboratories by people who are incredibly experienced at doing all of this. But there is a sort of strategy and an order, I think, that's important when you're telling the story. And I think that's perhaps a lesson that we've all learned about communicating science in the right way, through the right sort of outlets to make sure it's well accepted by the public.
Absolutely. So that was when we had H5N1, and then the next time you were largely called out, was that around swine flu?
That's right. So all of this sort of H5N1 story was going on in the background, various people trying very hard to understand why this bird virus was not leaping across into humans and causing the next pandemic. And then all of a sudden in 2009, we were all surprised, I think it's fair to say, that a virus came out from pigs and caused a pandemic, which travelled around the world very fast, started off in the sort of American continent, but of course within a matter of weeks, the WHO had declared a pandemic. And this is very scary to people because this is not in a lab closed away, this is out there in the street and something that can happen to everybody.
Yeah. So I think following on from that, there was an awful lot of work you were doing. This is when you were called out and you were literally having to shoot all over London. Yes. So what were the kind of interviews that you were doing then, and what were the kind of events you were getting involved in?
So I think in the early stages of any outbreak, what people want to know are the facts, as best you can tell them, which is why having people in my group sort of surfing all the possible routes of information and feeding that through in some digestible manner was important. One of the things I particularly remember doing was actually an interview with the children's presenters from BBC Newsround. And again, I mean, if you think about children in this kind of situation, they get quite scared. There's this new virus, they don't really know what it is, they're at school and everybody's talking about it. And so what the Newsround people particularly wanted me to do was to put on my white coat so that it would look as if I knew what I was talking about. And then, you know, in a calm way explain the situation, that most people that were getting infected by this virus were not getting very sick at all, but nonetheless it was good to be aware of this and to tell somebody if you were worried about it or if you thought you had symptoms, of course, you know, tell somebody and act early. We also ran an online question and answer session for children who had watched the Newsround article, and then they could email in and ask me questions. And it's amazing, actually, children, you know, ask some very good and clever questions. We grossly underestimate them. But I think it is important that, you know, it underscores the importance of basically speaking clearly and honestly as early as you possibly can to sort of allay those fears.
Yeah. I think striking up that dialogue as well is always quite important. I think on the course at Imperial, they've always called that out, having that dialogue, the on even with children. And so what were the outcomes? I understand that from swine flu, there was an awful lot of research which matches that graph there.
Yes, absolutely. So this is the only graph that we've got with us to talk about. I mean, one of the amazing things about the swine flu pandemic is that, thankfully, a lot of good research has been done because of this outbreak in humans. This particular graph is an epidemiology curve of what was going on in the UK. And again, it pertains very much to children, which is why I thought it would fit very well here. So what happened is that we experienced swine flu in the UK sort of in May and June and July, and the numbers were rising, the media were reporting, and lots of people were going along to their GPs imagining that they had this virus. And then amazingly, in around about the third week of July, the numbers plummeted, and you can see that very clearly from the epidemiology curve here. And they stayed low through the summer, and then in September they came back again. And this timing absolutely correlates with the schools breaking up for summer holidays. And this, I think, was the final piece of information that persuaded people in the Department of Health and on JCVI that vaccinating school children may actually be a really good way of controlling influenza, not only in pandemics, but actually in seasonal flu outbreaks, because it's quite clear that school children are major spreaders of this disease.
Yeah, which is absolutely fantastic news, but it's not something I've ever heard in the mainstream at all. It's something that I had to go and kind of read away on. So now we have this new vaccine that's coming out, which I know very little about. So I understand that it's given intranasally to children, as opposed to the injected vaccines that we see in vulnerable populations, such as the elderly.
Yeah.
So what's different about this vaccine to the ones we had available previously?
Right. So you're quite right, Jamie, that the first thing to say is that this is a live virus vaccine. It's an attenuated virus which is sprayed up the children's nose. That's great because the children don't have to get an injection. They don't mind this little squirt up their nose. The virus itself is cold adapted, so the virus can replicate in the cooler temperature of the nose, but it cannot descend further down to the lung where core body temperature would prevent it from replicating. So it's a well tried and tested virus, and it's now being rolled out in schools across the UK as a result of that epidemiology that was discovered or described in the 2009. What's fantastic, and I think you're quite right, not many people really know that this experiment, if you like, is going on in our children at the moment, and there is some fantastic data coming out from Public Health England, for example, on how well this strategy is working. So that in certain geographical areas of the UK, the pilot areas, the vaccine is being given to primary age school children. And if you look at the curves of incidence of flu-like illness and of actually mortality to respiratory illness, you can see that in the areas where the vaccine has been piloted, it's lower, not only in the children who receive the vaccine, but also across the board in all age groups. So I think that really underscores that this epidemiology informed this new policy. It's a shame, I think, that it's not being heralded as widely as it might as such a success story, but it's very, very exciting to see how we can go from, the sad pandemic outbreak to actually doing something that helps every year against seasonal flu.
Is it likely to be a challenge then? Because if we're immunising these children for the benefit of adults and the elderly later in life, to convince people that their child needs to be vaccinated, and obviously they're anti-vax.
Yes, absolutely. I mean, one wonders whether or not this is behind the sort of relatively subdued reporting about the success of this so far. I think it's, as I say, it's best really, I think, to be honest with people. The children don't really need this vaccine. Most young, healthy children don't suffer a severe case of influenza, getting infected with seasonal flu, such that they absolutely require to be vaccinated and protected against that disease every year. But what we know is that if we vaccinate the children, we decrease the total amount of influenza virus circulating in the community, and then the vulnerable people, immunosuppressed, elderly, who either can't take the vaccine or whom in their vaccine doesn't work so well, those people are then protected in a way that is much more difficult to do through the more traditional routes of giving them the flu shot, as we've been doing for some time.
And so, I know it's particularly with the elderly, but what have been, to your mind, been the kind of barriers to uptake of the current vaccine that we have, or the older vaccine that we have in the elderly? Absolutely.
So I mean, the flu vaccine situation at the moment that we have in the UK is a really good way of explaining, or trying to explain to people, that there can be different types of vaccine. So the live attenuated vaccine, which is being squirted up the children's nose, is a real virus, and those children are actually getting infected by that virus, and they're making a good immune response to it. The other type of vaccine that we've given for decades in the UK is an inactivated flu vaccine, and that's recommended to be given for elderly people 65s and over, as well as those who are at risk from a severe outcome were they to catch flu. That particular vaccine doesn't have a very good take-up rate. In the elderly, we're getting better because people are being persuaded to go along to various vaccine days at their GPs and get their jab. One group of people who are particularly bad at taking up the vaccine are doctors and nurses. And of course, it's very important that group of people, the healthcare workers, do have the flu jab each year because they're going to be coming into contact with people who are very vulnerable to flu, and we don't want them passing on any flu that they've picked up in the community to vulnerable people that they might be caring for. A lot of resistance amongst doctors and nurses in getting this in certain parts of the country or certain hospitals. Sometimes one hears from nurses in particular that the flu jab gives you flu and therefore I'm not having it because I'll need a day or two off work and it's more important that I'm with my patients. And again, this sort of new introduction of a different type of vaccine for children and the older type of vaccine for adults is quite a good opportunity to remind people of that difference, that the vaccine that adults get in this country doesn't give you flu. It's an inert injection of a protein into your arm which persuades your immune system to make antibodies that would protect you. It's a completely different kind of vaccine than we're rolling out to children at the moment. The other thing just to say about the doctors is that what doctors say is, oh, well, I'm a doctor, and of course I wouldn't go to work if I had flu, and therefore I wouldn't give flu to my patients. And again, I mean, work that we've been doing in our group and other groups, but also epidemiology that came out during the swine flu pandemic really reveals that, as for many, many virus illnesses, a lot of people get infected by the virus and can pass their virus on, and yet they don't show signs of symptoms. And they can also be contagious before the symptoms kick in. So when you get infected with flu virus, the peak amount of virus that's coming out from one person is coming out very early on in the first couple of days, and yet the person might not start coughing or sneezing until day 3, 4, 5. So although it is fair to say that coughs and sneezes can spread diseases, and we should cough into our elbow, etc., as all the public campaigns say, the point is that you can be contagious to another person even if you don't know you've got flu. And therefore that's why it's so important that we encourage our doctors and nurses and everybody to take up the flu vaccine and control the amount of flu that they're passing on.
And so with the dawn of this new live attenuated vaccine that's being used in children, why is that being used specifically in children and not in adults?
Mm-hmm. Yeah, so that's quite a tricky question. It's to do with the immune responses and the way that flu works. Flu is a virus that we get many times over our lifetime. When you get your first flu infection, you sort of lay down a memory response, a cross-reactive immunity, which can protect you not from infection by a subsequent virus, but can temper how ill you would get the next time around. And so there was a fear with vaccinating children with the inactivated vaccine. The inactivated vaccine is designed to completely neutralize the virus, to sterilize you, if you like, against infection. And it's felt that children should not be given those vaccines, if at all possible, because it is important that children have a flu infection early in life to lay down that basic cellular immunity memory. The reason is that, as you'll know, I mean, when a pandemic comes around, we don't have a vaccine available for some 3 to 6 months after the new virus emerges from an animal source. And in that intervening time, children who get exposed to the pandemic virus need some level of memory response to help them in dealing with that infection. If we'd have sort of sterilized them from birth against any flu infection, then the chances are that they would fare very badly when a new virus came along.
So on the theme of medicines and pandemics, I suppose the other issue that's been well publicised has been the controversy surrounding Tamiflu.
Yes.
Which is quite an easy sell for newspapers because all they have to say is that your government spends an awful lot of money on something that might not work.
Yes.
So what's been your view of this?
Yes, so as a virologist, I think it was important to get involved in the debate about Tamiflu. So you're quite right, the government spent upwards of £400 or £500 million in stockpiling Tamiflu drug, particularly sparked by the early the fears of an H5N1 pandemic that might have emerged, and such a severe virus would have required plenty of doses of antiviral to be given out in that intervening phase before a vaccine becomes available in the early stages of the outbreak. Tamiflu is an antiviral. It acts against the virus, against a particular protein of the virus, the neuraminidase. It's, the design of this drug is beautiful science. It's all very logical. Scientists crystallized the protein and designed a chemical that fits perfectly and works. So it's a fine drug. When you use it in the laboratory, as I do, it sort of decreases the virus replication. And so I thought it was important as a scientist to make sure that message came across, because about 2 or 3 years ago, a controversy brewed up over whether or not the government were right to have spent all the money on Tamiflu. And this controversy was really sparked by a particular campaign group who are known as All Trials. Ben Goldacre, who writes for The Guardian, and also the BMJ publication were sort of very much behind this. And they were trying to persuade the large pharmaceutical companies to release all of their data that gets collected when a trial is being conducted for a new drug. That wasn't the case previously. The case was that only the data that went forward to the regulatory authorities for approval and licensure of the drug needed to be shown to a wider public. So, I mean, whilst as a scientist I am very much in favour of sharing all knowledge. I mean, that was absolutely behind my involvement in the gain of function and the idea, those experiments, the idea that knowledge should be shared. I'm absolutely in favour of sharing knowledge about clinical trials. However, the problem with this controversy was that Roche, in fact, the makers of Tamiflu, did share that data, and it was reanalyzed by the Cochrane Group, who then published a report sort of basically criticising the way the trials had been conducted very much, and sort of saying that this drug doesn't work. And what they meant when they said it doesn't work is that it shortened the length of influenza illness only by about a day. Okay, so, I mean, most influenza illnesses last in adults between 5 and 6 days. And so the fact that it was shortened by just 1 day, Cochrane didn't really feel was good enough, and they didn't feel that justified...
450 million pounds.
Yes, 400 million pounds. Okay, so, yes, it does seem like a lot of money. You're quite right. But the point is that the trials the conditions under which those trials were conducted were in the late 1990s and the early 2000s. At that time, seasonal flu was not really causing a lot of disease, a lot of illness. The particular viruses had been in humans many years, and they weren't causing very severe illness. And it's quite difficult then to imagine how much better an antiviral drug could actually be than shortening the illness by just the 1 day. So what I mean by that is that if you think about the timeline of a person getting flu, they catch the virus. For the first day or two, they don't know they've got it. And all this time, the virus is replicating in their body. And then by day 3 and day 4, they're beginning to feel ill. By day 5, they feel really sick, and they go along to their doctors and they get Tamiflu. And then rather than staying sick till day 7, they now get better by day 6. So that shortening of an illness by 1 day is about as good as I think you could ever expect an antiviral drug to do under those circumstances. Now, if that person had been given Tamiflu on day 1 when they first got infected by the virus, then that drug would have had the possibility of suppressing the virus in their body and doing a much more efficacious job. And in fact, we know that if you use these drugs prophylactically, in other words, if you give them to people before they get infected, they work extremely well. So I think the problem with the Cochrane Group coming out and saying, look, 1 day is no good, it doesn't justify all this spend, is that 1 day is about as good as you could expect a drug to do under the particular set of circumstances where those trials were conducted. On the other hand, in the pandemic, the swine flu pandemic, we did use Tamiflu, and we gave it to people who presented in hospital with severe flu illness. And retrospective analysis shows that strategy was very successful. It saved lives, particularly in high-risk groups like pregnant ladies. It made a huge difference whether or not they got Tamiflu early when they got to hospital, or whether that administration was delayed. So there are very robust studies with retrospective analysis showing that this drug really does work, it really does save lives in a pandemic situation when people are thinking about flu and they're presenting early in their illness, and doctors are thinking about flu and getting the drug into the patient fast enough. So in terms of pandemic preparedness, if a government were to make a decision that they weren't going to have a drug stockpiled, I think that would be a very tough decision for governments to make, and probably we need more information to help them make such a decision. I think what it's taught me about viruses and antiviral drugs moving forward is that developing antiviral drugs for viruses whose illnesses are very short-lived is quite a challenge. And actually, we have a good antiviral drug against flu, but we don't have a very good diagnostic. So most people don't know that they have got flu, or the doctor doesn't know that it's a flu case and they should give Tamiflu early. So I think one of the things that comes out of all of this is that by communicating science and thinking through the issues and working out where the problems lie, you can actually identify that it's not really the antivirals that are lacking here, it's how we use those antivirals.
Yeah. Sure. I think we had some examples coming at us about your experiences with science communication beyond this, and there's a particularly good example through this crystal glass version of flu. So what happened with this, with the glass flu?
Yes. So this is a really beautiful sculpture which you can go and see in the Wellcome Trust building in Euston Road in their collection. One of the best experiences I've had doing science communication was I was fortunate enough to be on a television program that was made a couple of years ago. It was called Winter Viruses and How to Beat Them. And it featured Michael Mosley and Alice Roberts, and it was talking about flu, and we were trying to find somebody on the street who had flu and diagnose them in real time and talk about lots of issues. And part of the setup, the crew had got this beautiful glass sculpture on loan from the sculptor himself, and it was sat in the middle as a sort of centerpiece, and the rest of us were sat on a table a bit like this, sort of discussing viruses and what they're made of and how we can tell if we've got them. And we had been told at the start that this sculpture was worth thousands of pounds and that we must be incredibly careful about it. But as the day wore on and we were on the fourth take of Alice trying to ask me to describe a virus or whatever, the various cameramen were sort of bustling about and the sound people with all their gubbins on their back. And one of these poor sound people with a great big rucksack of kit on turned around and knocked this sculpture flying, and all of a sudden this flu virus was broken into 1,000 pieces on the floor. And of course everybody sort of gasped and was absolutely horrified. And of course it was awful because it was a wonderful sculpture, but it was also awful because we were getting towards the end of a day's filming, and one of the things I've learned is about continuity. You will know this. So basically the problem was that if we couldn't find another one just like it, we'd have to shoot everything all over again because the sculpture was in all the various shots. And amazingly enough, we all went home that evening, and the next day another one of these sculptures had appeared. I know not where they managed to get it from, but I think it illustrated to me that people in the media make things happen when they need to happen, and there was this glass sculpture all over again, and this time the sound man was allowed nowhere near it.
That's right. After our YouTube interview with you, I had to reshoot a whole bunch of things without you there because the camera that had been focused on me was out of focus. It was really difficult trying to interview like an empty chair. It's very, very challenging. Okay, so in terms of science communication generally, do you feel now that it's becoming the role of scientists to dispel these kind of myths and step up really?
Yeah, I mean, I think the point is that we ask the public to take their children along to be vaccinated. We ask them to allow us to spend taxpayers' money on drugs. So we have to explain that to them. We have to explain it in a language. We have to face up to these facts. If we think about various scientific examples like GM crops, etc., where scientists didn't speak up soon enough, we end up with a problem. And we can't expect the public to embrace our science with the enthusiasm that we embrace it unless we step up to the mark. I think there are so many young people out there today that see that already. As I've already said, there are many, many people in the audience and across the Microbiology Society who do this. But if there are other people out there who are tempted to have a go, then really the fantastic route in is to contact the Science Media Centre because they run a list of experts. And the point about microbiology is that it impacts on lives in many ways. Vaccines, pathogens, sort of all kinds of things that we do in everyday life in microbiology need translating to the public. So if there are budding microbiologists out there, and if there are aging microbiologists out there who feel like getting more involved, in all of the communication, then I think the Science Media Centre is a fantastic route forwards. They run classes, they will put you on their list, and they will contact you if something comes along that you are needed to comment on.
Sure. Well, I mean, I ran out of questions, but thanks very much.
Thanks very much. And thank you to Jamie for volunteering to come and interview me. And thank you very much to the Society for listening to me and for the prize.
Thanks.